Dear audience, dear Chairman, Professor Kotelevtsev, we have the quorum of the jury, and as soon as we're ready, we can start the procedure.
Okay, thank you. I'll start as usually with introduction of jury members, and first to the left from me, Mikhail Gelfand, Professor of Skoltech and the Director of the Center of Molecular and Cellular Biology and Vice President of Biomedical Research, graduated from Moscow State in 1985 and has worked in the field of bioinformatics, molecular evolution, earning a candidate of science degree in mathematics and doctoral degree in biology. He is world-known specialist, and his knowledge encompasses comparative genomics, systems biology, molecular evolution, and many others. He is elected member of Academia Europaea and a member of the editorial boards of multiple peer-reviewed journals, and regularly serves on the program committees of international workshops and conferences on genetics, biophysics, and bioinformatics. Then Edze Vestra is a professor of microbiology at the University of Exeter. His lab studies encompasses ecological variables, drives the evolution of various immune strategies in bacteria with the emphasis of CRISPR-Cas, adaptive immune system. And he is a member of the Environment and Sustainability Institute and the Bioscience Department of the College of Life and Environmental Sciences. After completing his PhD in the lab of Professor John van der Oost at Wageningen University on the molecular mechanism and regulation of CRISPR-Cas immune system, Edze Vestra started to explore the evolutionary ecology of CRISPR-Cas immune system as a Marie Curie Fellow in the lab of Professor Angus Buckling, the University of Exeter, received a NERC Independent Research Fellowship in 2015 and set up his own lab in the University of Exeter. Edze Vestra has received several prestigious awards, including being a finalist for Blavatnik Award for Young Scientists in UK in 2020-21 and the Fleming Prize for the Microbiology Society in 2020 and Philip Leeuwenhoek Prize in Biological Sciences in 2020. Next jury member is Olga Suturina, University Paris-Saclay, France. Olga is professor at Paris University, Saclay University, and an associate professor in the Department of Microbiology and Infectious Disease in the University of Sherbrooke, Canada. She is also the co-supervisor of Fundamental Microbiology Master 2 program and AGR advisors in genetics, genomics and microbiology at the Graduate School of Life Sciences and Health, University Paris-Saclay. Professor Suturina serves as the Deputy Director of the Microbiology Department at the Institute of Integrative Biology of the Cell. She leads the regulatory RNAs and Clostridia laboratory. Her research focuses on the role of non-coding RNAs in Clostridium difficile, physiology and regulatory networks, particularly in the pathophysiology of this major human enteropathogen. The group also investigates the CRISPR-Cas9 systems of C. difficile, examining the function and regulation. Next jury member is Associate Professor Raymond Stahls, Wageningen University, Netherlands. Raymond Stahls is the Associate Professor at the Wageningen University. He completed his PhD in Biomolecular Chemistry and Radiology Institute for Molecular Life Science in Netherlands in 2011. From 2011 to 2014, he worked as a research fellow at Wageningen University in the Laboratory of Microbiology, and from 2014 to 2016, He worked at the University of Otago, New Zealand, in Professor Peter Finneran lab and the bacterial adaptive CRISPR-Cas immunology system. Dr. Stiles works on the project titled Carving Out of the Signaling Network of CRISPR-Cas, where the newly discovered CRISPR-associated signaling system is explored, along with its multitude of enzymatic activities and the repurposing for genome engineering. He is a co-founder of Scientific Advisor and a board member at Scope Bioscience, a company that aims to provide innovative and accessible CRISPR-Cas9-based tests for infectious diseases and genetic trials. The last jury member is Dr. Robert Fagerlund, Research Fellow, University of Otago, New Zealand, Department of Microbiology. Working in Professor Peter Finneran's lab, his main goal of the research group is to understand the interactions between mobile genetic elements, such as bacteriophages and plasmids, and their bacterial hosts, with primary interest in phage resistance mechanisms. Robert Fagerlund completed his PhD in biochemistry and molecular biology at the University of Otago in 2009 and worked as a postdoctoral fellow at the biochemistry department. He then continued his research as a research scholar at Penn State University from 2012 to 2014. He has deep interest in CRISPR-Cas biology, from understanding its natural defense mechanisms against viral infection to leveraging its potential for new biotechnological applications. Dr. Fagerlund probably won't be able to join us, but I will read his review at the end. And finally, the supervisor of Thesis is well-known Professor Konstantin Severinov. He is professor at Rutgers and the head of the laboratory at the Waxman Institute of Microbiology. He leads the Laboratory of Molecular Genetics of Microorganisms at the Institute of Gene Biology and the Russian Academy of Sciences. Konstantin Severinov graduated from Moscow State in 1990, received his PhD in 1993 and a doctoral science degree in biology in 2006. His research interests include microbial natural products, defense systems of prokaryotes, molecular biology of bacteriophages, mechanisms of gene regulation, and metagenomics of natural microbial communities. He has co-authored more than 200 publications in peer-reviewed journals and holds 8 patents. And now Karyna Karneyeva. She received a specialist degree from Belarus State University Faculty of Biology in 2017, and the same year joined Caltech for Master Science program. Since then, she has been working under supervision of Professor Konstantin Severinov on their projects related to bacterial defense system, particularly CRISPR-Cas system. During long-term academic mobility at the Pasteur Institute, she worked in Mart Krupovich's lab on a study related to viral infection processes in extremophiles. In 2019, Karyna entered the Caltech program in life sciences. During her PhD studies, Karyna has co-authored 3 publications and participated in international conferences. This is all for presentations and short reminder for the procedure. We have 40 minutes limit of your presentation, then questions and answers, and we encourage questions from the audience and questions from the Zoom audience as well. It's important that each jury member confirms whether he is satisfied or not with the changes introduced to the thesis after the review. Questions from general audience, okay, this is what important statement. And okay, so I'll come later to the later stages, but now I invite now our defendant, Karyna, to present her thesis. It's all yours.
Dear Professor Katilevtsev, thank you very much for the introduction, and good afternoon, everyone. Good afternoon, dear jury members, colleagues, and friends. Thank you very much for coming. And today I'm glad to present you my PhD research project devoted to the exploring type 3 CRISPR-Cas immunity in Thermus Thermophilus that was conducted under the supervision of Konstantin Severinov. So now it's working. I am working on type 3 CRISPR-Cas immunity, adaptive bacterial immunity systems, and the focus of my research is type 3 systems. They stand out because of the complexity of the immune response, and there are lots of unclear features that we would like to know. Still, there are three main stages for type 3 CRISPR-Cas immunity, and the first one is the adaptation, the insertion into the CRISPR array of new spacers. The second one is maturation, the transcription of a long pre-CRISPR RNA that further can be processed by Cas6 protein, and small CRISPR RNAs are formed. And the third stage is interference, and in the case of type 3 CRISPR-Cas systems, it has several unique features. And first of them is a transcript rather than the DNA. And when the effector complex recognizes the target, three distinct activities might be activated. So the first one is specific RNA cleavage. The second one is non-specific DNA cleavage, and synthesis of second messengers, cyclic oligoadenylates, that further can activate different auxiliary proteins that possess DNAs, RNAs, and other activities. Another distinct feature of type 3 CRISPR-Cas immunity is the way of autoimmunity prevention. There is a 5' tag the complementary region of the invader. All these activities can be activated. And the model organism of our research is Thermus Thermophilus, and on this slide you can see 4 CRISPR-Cas systems that are encoded in its genome. Its genome consists of a megaplasmid and a chromosome, and there are 2 type 3 CRISPR-Cas systems, 3A and 3B, and both of them share the same CRISPR arrays. So here you can see the thesis structure, and the results are summarized into chapters where we try to answer 2 main questions: type 3 CRISPR-Cas immunity of Thermus Thermophilus against phages, and the second one is principles governing target recognition by the type 3 effectors. So let's move on to the chapter 1, and again, here you can see the genome of Thermus Thermophilus, and to differentiate the activity of type 3A and type 3b systems. Two mutant strains were obtained in our laboratory previously. The first one is 3A plus strain with deactivated 3B module, and 3B plus strain with deactivated 3A module. And to test the immunity of these systems, we used the following experimental system. Double-stranded RNA fragments that included type 3 repeats and desirable spacer were inserted into PMK18 vector, and then thermostromophilus cells were transformed with such constructs. And these strains further was tested to the ability to withstand phage infection. So here you can see that the strains, wild-type strain that has both systems activated, type 3A plus strain, type 3B plus strain, and a strain with both systems deactivated that were tested after they were transformed with an empty control plasmid without spacers. And you can see that there are several dilutions that was spotted on corresponding lawns, and phage in the case if these strains bear empty plasmid were able to form plaques, meaning that these strains are sensitive to phage infection. But if we transform wild-type 3A plus and 3B strain with a plasmid bearing space targeting transcript from FIFA phage genome, we can see that such strains become resistant, and only strain with both systems deactivated is sensitive to the phage infection. The same results were obtained for the phage FICA, and only wild-type 3A and 3B strains bearing a plasmid with a space corresponding to the transcripts from the gene 25 were resistant to phage infection. Further, we tested a set of plasmids and used the same construct with spacers that targeting FIFA genes corresponding targeted transcripts from early, here is E, middle, M for the middle construct, and late constructs. This early, middle, and late means when the phage starts its transcription during the phage life cycle. And we showed that only spacers corresponding to the alter region protected cells from FIFA infection. But still, we had to prove if other spacers that we tested were functional. To test it, we created another plasmid-borne structure with a mini CRISPR array and corresponding protospacer. In the case the protospacers trigger, the transcript of these protospacers trigger the immunity, then these cells cannot survive on the selective media. And indeed, we see that if the spacers correspond to transcripts from early, middle, and late genes were able to trigger the immunity, meaning that in fact these spacers are functional. And it's important to know that all these spacers that we tested, we chose because of their presence in the after the native adaptation we observed in our lab previously. So we repeated the same procedure for the FICA phage, and we see that spacers, I'm sorry, the clicker. We see that spacers targeting transcripts of all classes, early, middle, and late, can protect cells from FICA infection. And this is in contrast to the FIFA phage that I described previously. So we hypothesized that FIFA alter genes may encode some anti-CRISPR proteins inhibiting type 3 immunity. Further, we monitored the dynamic of the growth for infected cultures, and on the slide you can see growth curves for strains without spacers, meaning that they are sensitive to the phage infection, and they were infected with the phage and lysed shortly. But in the case of the strains bearing protective spacers, such cultures continue to grow. And the same trends we see in the case of FICA phage infection. So we concluded that thermostromophilus cultures with spacers targeting phage transcripts survive the infection, meaning that in our case, type 3 immunity doesn't work in a suicidal manner. However, we noticed that in the case of FICA, all infected resistant strains continue to grow in the same manner as non-infected wild-type strain. But in case of FIFA, we can see a decrease of the growth rate, and we decided to check the overnight cultures for these strains and spotted lysate obtained on wild-type strain or these resistant strains bearing spacers against gene number 42 or gene 44. And we see that all of these supernatants, in fact, had viable phage particles, and they can proliferate. But if we check the same lysates on the previously resistant strain, initial strain, then we see that this strain is still resistant to wild-type lysate, but we see plaques if we spotted lysates that were obtained from the same strain, and the same for the strain bearing spacer against gene 44. We isolated several phages from this and this plates and performed full genome sequencing, and here you can see the alignment of the wild-type phage genome and sequences of the escapers. And we concluded that when we target essential gene, for example, gene 44 is essential since it is RNA polymerase gene, The phages can escape type 3 immunity, accumulating deletions of an integral number of codons in the protospacer. But if a non-essential gene is targeted, longer deletions of various size are accumulated in the targeted protospacer region. So here you can see the main conclusions for the chapter 1. We showed that both type 3A and 3B CRISPR-Cas systems protect Thermostomophilus from phage infection. Also, Thermostomophilus cells with protective spacers that target phage transcripts survive the infection. Thermostomophilus strains bearing plasmid bone mini CRISPR arrays with spacers targeting protospacers in early but not middle and late genes transcript of bacteriophage FIFA are resistant to infection, while Thermostomophilus strains bearing plasmid bone mini CRISPR arrays with spacers targeting all gene classes of bacteriophage FICA are resistant to infection. Also, deletions in and across the phage protospacer regions that result in multiple mismatches with protective type 3 spacers lead to the escape phenotypes. So let's move forward to the chapter 2 that is devoted to the interference requirements of type 3 CRISPR-Cas immunity. And in this case, we used the following experimental system. Type 3A and type 3B CRISPR-Cas systems can decrease transformation efficiency of plasmids. And to test this immunity in detail, we used two variants of PMK18 constructs, and in the case of the PSDIR, plasmid PS is for protospacer, we inserted a protospacer into the vector. This protospacer corresponds to the spacer from a genomic CRISPR array. And in the case of PSD, the transcription of this region goes from the PSLPA promoter. It is a strong constitutive promoter, and when the CRISPR RNA target such transcript, it can mediate the immunity, preventing colonies transformed with such plasmids from growing on selective media. And in the case of PSREF plasmid, the transcript from a PSLPA promoter is identical to CRISPR RNA, and that's why the antisense transcripts are recognized, and according to our RNA-seq data analysis, the abundance of such transcripts from PSDIR plasmids is 80-fold higher than in the case of PSREF plasmids. So both type 3A and 3B systems could decrease transformation efficiency on plasmids. You can see the 10 microliters serial dilutions of the transformant mixtures that bear a plasmid without protospacer, and meaning that such plasmid can transform efficiently. But in the case of PSDIR and PSREF, colonies cannot grow on the selective media, meaning that type 3A and 3B immunity is efficient against plasmid transformation. However, you can see that there are some colonies that still can grow on the selective media, and we isolated such colonies, extracted DNA, and performed sequencing. And turned out that some colonies formed after transformation of cells mounting type 3 immunity contained extensive deletions that result from recombination between type 3 arrays repeats. So on this Zucker's plot, you can see the megaplasmid sequence, and in the inner circus, you can see in black CRISPR arrays, here, here, and here, and also Cas operons that correspond to type 3b system and 3a CRISPR-Cas system. So in the case of escapers for 3a plus strain, deletions that provide the absence of 3a module were observed. And these blue lines connect borders of the detected deletions. And in the case of 3b system, the same, that deletions of type 3b module occurred. And the biggest deletion was the same for 3a and 3b strain, led to the both operon absence. So we concluded that type 3 CRISPR repeats are recombinogenic, and this is the first time noticed phenomenon. So coming back to the requirements for target recognition by type 3a system, we performed again, transformation with different sets of plasmid derivatives. Such derivatives bear mismatched regions, and they are labeled according to the maximally expected lengths of the duplex. And if such duplex can lead to the immunity, then it's colored in black. And if we don't see interference and the cells can grow on the selective media, such duplexes is in magenta. So we tested different variants of sets for PS0 and PSREF plasmids, and here you can see structures, schematical structures of all effector complexes with maximally mutated duplexes that can provide resistance, that can provide interference for 3A and 3B systems. And there are also high level of target transcription and low level of target transcription. And up line correspond to the mismatches introduced to the 3' end of CRISPR RNA. And on the bottom, we can see duplexes with mismatches at the head of the duplex. And you can see that in the case of type 3b immunity, we see that such duplexes must be longer. That means that type 3b systems are less tolerant to the mismatches, and 3a systems are more effective. But these differences might be explained by the diverse features of type 3A and type 3B system, and one of them is the presence of an HD domain, in the Cas10 subunit in the case of type 3A systems. This subunit is responsible for the single-stranded DNA cleavage, and this domain and type 3b system lacks this HD domain in its Cas10 subunit. So we decided to check if there is any impact of this domain into the type 3A immunity. To check it, we constructed a strain with a mutated HD domain, and surprisingly, even the fully complementary duplex was not able to provide the efficient interference, and such and cells transformed with these plasmids were able to grow on selective media. That means that in fact HD domain has a crucial role in type 3A immunity. Still, we can see that these cells are smaller, and that means that still there are some interference that is triggered by the recognition of the protospacer. And our hypothesis was that these cells has smaller sizes because of the smaller plasmid per genome ratio, meaning that plasmid just cannot be eliminated from these cells. And we performed qPCR, and indeed, in the case of HD mutation, the plasmid per genome ratio is drastically lower than in the case of wild-type HD domain. So we concluded that the HD nuclease activity is crucial for efficient type 3 immunity against plasmid transformation. And the absence of HD nuclease in type 3b effector suggests that it relies on some alternative mechanisms of interference. Another interesting thing that we found is the way actually, we decided to try to introduce mutation at both ends of the CRISPR-RNA target duplexes. And we revealed that there is a pattern, and there is a minimal there is a minimal RNA duplex that is sufficient for interference, but if we introduce more mutations at the 5' end of CRISPR-RNA, such mismatches can be the effect of these mismatches can be abolished if we add more complementary base pairs at the other end of the duplex. And we concluded that there is a minimal RNA duplex that is sufficient for the interference, and this RNA duplex can occupy multiple positions within the type 3A effector. And based on this, we assumed that there is a correlation between efficient type 3 immunity and the energy of binding between the duplexes and the effector. We tried to propose a model for this observation, and the model was developed by our collaborator Yaroslav Ispolatov from University of Santiago in Chile, and he made the several assumptions: that the binding of the effector and the target transcript is reversible, that the stationary concentration concentration of the effector-target complex is determined by the mass-action equilibrium, and the dissociation constant is expressed as the exponent of the Gibbs free energy difference of the effector and the target transcript. So here you can see the final equation for our model, and it suggests that the lower level of target transcription can be compensated by the longer duplex. And we decided to check if it's true for our experimental data. And as I mentioned, in the case, based on our RNA-seq data, in the case of high level of target transcription, it is the abundance of the transcript is 80 times higher than in the case of low level of target transcription. And if we use these values for this equation, the difference between required complementary nucleotides in this case and this case will be almost 3. So if we come back to our slide with general results, we see, for example, let's look on the type 3A system. In the case of high level of target transcription, the minimal required duplex is 23 base pairs, but in the case of low level of target transcription, it is 26, and it's 3 base pairs that indeed is required for efficient immunity in the case if we have less abundant target. So conclusions for chapter 2 are the following. We show that Thermus-Thermophilus type 3 CRISPR repeats are recombinogenic. Also, we show that length of RNA duplex required for type 3 CRISPR immunity in Thermus-Thermophilus depends on the target RNA abundance. And also, we show that HD nuclease activity is crucial for efficient type 3 immunity, and the absence of HD nuclease in type 3b effectors suggests that it relies on some alternative mechanisms of interference. Also, type 3 effectors bind their target through a simple bimolecular reaction where extensive CRISPR-RNA target base pairing compensates for lower target abundance. And the minimal RNA duplex sufficient for interference can occupy multiple positions within the type 3A effector. Taken together, our research revealed some intriguing insights into the type 3 immunity and created a base for future research. Particularly, we are interested in studying known and predicting new auxiliary proteins that can have some contribution to type 3 immunity. Also, it's interesting to investigate type 3A or 3B CRISPR-Cas immunity against newly discovered Thermus-Thermophilus phagers and exploring anti-CRISPR proteins inhibiting type 3 immunity in Thermus-Thermophilus. And actually, I think that Thermus is a great model organism to dive into the type 3 immunity details. So the results of my work were published in 2 articles and presented on 2 conferences. And so I would like to come to the acknowledgements. And first and foremost, I would like to express my gratitude to my supervisor, Konstantin Severinov, for his guidance, for his expertise and mentorship during all my research. Also, I would like to express my appreciation to Daria Artamonova, who introduced me to this field and provided continuous support. And also I would like to thank Matvey Kolesnik for his input in this research. And also I would like to say thank you for all my current and former colleagues at Institute of Gene Biology and at Skoltech, and our sense of community made this journey much more richer. Thank you all. Yes, this is it. Thank you for your attention.
Thank you very much, Karyna, for being on time. I think it was very clear presentation, and our custom is that each of the reviewers examines you, asks questions, and you reply to them. And to make it consistent, it's just one reviewer at a time. And we start in the order of appearances with Professor Gelfand. His questions.
Well, with me, it's relatively simple because all my questions have been addressed during revision of the manuscript, so I don't see a need for any further discussion on that point, and I'm completely satisfied with the responses I got.
Great, thank you very much. So then we come to Edze Vestra. Is Edze with us? Hi.
Hello, hello. Thanks, Karyna, for the presentation, and already, congratulations on your completing your thesis. It's a very interesting piece of work. I enjoyed reading it. So I would like to, to discuss some of the data that you present in chapter 4 in more detail. So I guess I had, some questions around the specificity towards the LTR region, and you provide some explanations, but in the chapter you don't really arrive at a very solid conclusion why this particular region is being more effectively targeted over the other regions. So maybe we can, first of all, maybe you want to elaborate on what you wrote in the thesis and explain your thoughts as to why this particular region may be more sensitive to targeting. What are your current thoughts on that?
Thank you very much for the question. And yes, I have several thoughts about it. So I think that this region, can you hear me? Sorry.
Yeah, I can hear you.
Okay, great. So the main point that I would like to mention, that it was shown in vitro that the recognition of base pairing at the 5' end of CRISPR-RNA influences on synthesis of cyclic oligodenylate drastically. So there are some structural arrangements that happens if there is this base pairing, and there was shown that exactly the same positions that we discussed are important for this for the synthesis of second messengers, and since type 3 immunity relies majorly on the auxiliary activities of different proteins, that means that this region indeed important for the efficient immunity. But in the case of mismatches from the tail of the effector, we couldn't say anything solid, because we know that in vivo these complexes can vary in the amount of subunits that form it. So that's why it's quite difficult to analyze these data and made any assumptions.
Okay, I'm not sure if I completely follow the reasoning here. So we are talking about, for example, figure 4.4, where the protospacers within the LTR region are associated with higher levels of immunity compared to the protospacers outside of that region. And it wasn't clear to me just now why you would get different activations when...
Oh, I'm sorry. I thought that we are speaking about the chapter about interference requirements.
Okay, no
Maybe
problem.
I
No
just
problem.
misunderstood you. So, yeah, can we go please to the slide with the space, I can... the one with the FIFA-phage genome and... I don't remember, sorry. So can you just turn on? Yeah, and I will switch to the right one.
Okay, so yeah, I'll just repeat my question in the meantime.
Yep.
So, the question was, why are protospacers in that particular region more effective compared to the others? And in your thesis, you have a couple of explanations. One of them is that there may be some anti-CRISPR activity encoded within this region, and therefore neutralizing the phage very early on might be advantageous. Are there, in the meantime, additional hypotheses that you wanted to share?
Thank you. Can we move to the previous slide, please? No, now it's working. Yeah, thank you. So yes, there are actually different variants to explain why particularly this region is important. And as I said, one of them was the most interesting, let's say, for us. We expected to find some anti-CRISPR proteins in this region, and we hypothesized that maybe synthesis of these anti-CRISPRs will lead to inhibiting the type 3 immunity. But there is another explanation, more ordinary, that some proteins of the phage may had some toxic activity on the cell metabolism, and that's why even if the other spacers provide any resistance, the cell cannot recover from the toxicity of the phage. And also one more interesting, let's say, variant of to explain this thing is the idea that there is a gene 44 that encodes viral RNA polymerase, and since we know that type 3 immunity depends on the transcription, there might be some differences between the transcription with single subunit RNA polymerase of the phage and a bigger polymerase of host, and maybe there are some specific interactions in the transcription elongation complex in the transcription bubble. So maybe there are also some hints to answer the question that relies here. So.
Okay, yeah, so that makes sense. So a couple of different possibilities. And so if we delve a little bit deeper, then if you look at those different protospacers, you don't see differences in plaque numbers, which is one indication that they may not be very effective. But then maybe there's other metrics that you could look at as well that could provide some additional insight whether these protospacer targets are doing anything, or whether the corresponding spacers are doing anything. For example, the phage burst size.
Yes.
Have you looked at that?
We haven't looked at this, but what's going on with the presentation? So, for example, in the case of the plasmid bearing spacer, matching the transcript from the gene 36, you see that there are some plaques, in fact. So this is kind of a partial effect that would be interesting to investigate. And also, spot tests help us to see the propagation of the phage. But maybe there are some kind of suicidal strategy that is taking place for other spacers where cell dies but cannot provide effective, cannot provide propagation of the phage. So actually there are so many interesting details that can take place, and I think that first of all quantitative analysis will help us, and also we could measure phage burst effect to see maybe some other interesting insights.
Right. Is this phage injecting its genome in one go, or is it a two-step process?
It's injection.
And the whole genome is injected at once?
So we know that, I'm sorry. So we know that the long terminal repeat region is the first region that the region that is injected first, and also maybe there is a hint for the revealing the answer for your first question, that maybe since this region is injected firstly, so mainly there is just more time for immunity to protect cell.
Yeah, that sounds like a plausible explanation. How different is this then from, you know, what Josh Modell reported previously with, you know, the Streptococcus system? Do you think the mechanism could be, you know, very similar?
Can you please repeat?
A couple of years ago, Josh Modell from the lab of Luciano Marafini published a paper where they found preferential acquisition from, you know, the regions that are injected first. So I was just asking you whether you think that then, you know, the underlying mechanisms are comparable, or whether you think there's some fundamental differences still.
Yes, so this is, I think you mean the latest, not latest, but recent paper from them. So yes, they show that in fact HD domain, about what I was spoken about, that is active in type 3A system, that when there is the activity of HD domain, the cells can recover from the dormancy. And actually, since we know that type 3B system likes this domain, maybe indeed just these spacers cannot allow the cell to recover from dormancy, and that's why these spacers don't lead to the survival of the culture.
I see. Okay. So then, you know, talking about the anti-CRISPR hypothesis, which is an interesting one as well, but, you know, in the end, you didn't find any convincing evidence of anti-CRISPR activity in this region. To me, it was a bit of a black box how you identified your promising candidates to start with. You mentioned in the text that you used a particular tool, but what made you, you know, convinced that these would be anti-CRISPRs? What particular characteristics were you looking for?
So you mentioned, you mean the part about anti-CRISPR proteins that we, what?
Yeah. So you described that there are 3 strong candidates, or 42, 45, and 47, that might well be anti-CRISPR genes. And you know, you don't explain why these are strong candidates. So I'm asking, why did you think they were strong candidates?
Actually there was a pipeline described by Eugene Koonin and his team. There was an article about how they try to predict novel anti-CRISPR proteins, and we in fact asked them to help us and maybe use the FIFA genome to reveal some anti-CRISPR proteins. And they used their pipeline and predicted that there are 3 candidates that are not iron 1, but maybe they have some activity. And so that was just the... Can you hear me?
Yeah, of course. Yeah.
So, yeah, great, thank you. Yeah, so yes, that was a machine learning approach that was described by Eugene Koonin. So actually, we don't. I cannot say what exactly was in the prediction.
But when you looked at those 3 genes, those were your candidates. Did you look at how conserved they are? Whether you find them in other mobile genetic elements? Whether they are maybe associated with known anti-CRISPR genes? Did you do these kinds of analyses?
I'm sorry, we had some interruptions and I couldn't hear your question.
Right. Okay, so I was saying, once you had your 3 candidate genes that might have anti-CRISPR activity, did you then, look whether these genes were also present in other mobile genetic elements, maybe whether they co-occurred with known anti-CRISPR genes, these kinds of analyses? Did you try that, and did it, strengthen your hypothesis that it could be a counter defense related gene?
These genes are very unique because many viral proteins, in fact, are unknown and have some putative activities. We didn't find any homologs to them, and I know that searching for anti-CRISPR is not an easy thing because all occurs are very diverse. That's why it's not surprising that we couldn't see something familiar to our proteins.
Okay. So they're unique. And do you still think they are anti-CRISPR, maybe against other CRISPR-Cas types?
We haven't checked it, but this direction regarding all these predicted candidates and maybe other genes from alter region would be of high interest for us, and I hope that we will find something interesting here.
Yeah. So what we found in the past is that when phages have anti-CRISPR genes, if you throw high enough doses at the bacteria, they will amplify. But when you throw low doses, they won't amplify. Have you looked at that? Whether you see phage amplification if you throw lots of phage, but no phage amplification when you throw, when you use low doses? Have you tested anything like that?
The problem is that we couldn't obtain the high concentration of FIFA phage at all. The maximum was 10 in power of 9, I guess. So we couldn't check it in a liquid culture, for example. But if we use, this. Sorry, we have some interruptions every time. So, for example, in the case of FICA, we see some spots that are not plaques, in fact, but some decrease of the growth, and maybe we could see some trends that you are speaking about if we could use higher titers.
Yeah. Well, I've got a couple more questions, about this, and then I'm done. But I did wonder more generally, what happens to your phage? So you are
We lost
showing
you.
a population. Sorry?
Hello. We lost connection to the microphone.
I'm sorry, we lost connection, I guess.
Okay, so I'll repeat
Due to your
what I.
questions, we lost communication.
Okay, so shall I repeat the question?
Yes, please.
I was saying I just have a couple more questions and then I'm done. So that's good. But my one question I had was more generally around sort of what happens to the phage. So you showed plaque assays, but you also did some population dynamics experiments. So they are shown later on. Which figure is that? It's figure figure 4.14. You show the dynamics of the bacterial population, but you don't show what happens to the phage. So what happens to the phage here? Is it going extinct or is it persisting?
So you're speaking about these images, yes?
Yes.
Oh,
Yeah, that's right.
oh, I guess we've lost connection again. I'm very sorry for this. But I asked if you're speaking about these images.
Yeah. Yes.
We actually, at the very beginning of my work, I checked the burst size of the phage FICA, and indeed that was a drastical decrease of phage particles in the supernatants. But I haven't checked it for FIFA, and I think that these phages, in fact, are very distinct, and we should not use the things that I saw for FICA to tell about FIFA, because we see that, for example, FICA phage cannot escape, for example, immunity, while FIFA can. And also in the case of FICA, we see survival in the case of strains bearing spacers targeting different transcripts. So unfortunately, no, I haven't checked it for FIFA, but my colleague works on these things using strain HB8, and I hope that soon we will hear some new interesting results regarding this question.
Yeah, no, absolutely. I think that would be very interesting also because these phages do behave, so differently as you were just mentioning. So it would have been nice to see, the population dynamics, not just of the bacteria, but also of the phage, and to maybe map that onto the evolutionary responses that you see. So I guess I have two final questions. So one is there any spacer acquisition during those assays? Do you know? Did you check for that?
Spacer acquisition, actually, the first paper where these results were published were devoted to the adaptation, naive adaptation in Thermus Thermophilus under laboratory conditions. So yes, there is adaptation that we actually can check and observe in laboratory.
Okay, and that's against both phages you see during this experiment over this time scale of just, 180 minutes, you see a lot of spacer acquisition happening.
We haven't checked it particularly because to the adaptation test, we used a very low multiplicity of infection and incubated samples for a long time, made re-inoculations. So unfortunately, we didn't check the changes and the spacer acquisition in these particular strains after all.
And then my final question for you is, how do you explain that Phi-Co is unable to evolve to overcome CRISPR-Cas immunity, whereas Phi-Fei can?
I think that the thing is that ficphage, as we can see here, is a small virus that has only 26 genes. That means that all of them are quite important for the life cycle, even if we don't know the particular function. So maybe, as we see in the case of FIFA, the deletions should be so accurate that we just cannot see this rare this rare event in our cultures, but maybe more attempts will help us to detect something. But I think that it's just some viral biology question, and that's it.
All right. Well, thank you very much.
That's great. Thank you very much.
Thank you very much for the discussion.
Yeah, the
Very interesting
discussion
questions.
was very interesting to follow it indeed. So we go to the next jury member, and this is Olga Saturina. Olga, are you with us? Yes. So Olga, so please go through the examinations, and probably I need to set a limit of maybe 20 minutes for the discussion with each reviewer, because otherwise we will be here late after our schedule. Please, go ahead.
Well, I spent about half a minute, so I present my remaining minutes to Olga.
Okay, thank you very much.
Yes, I will keep for further members. So thank you very much for invitation. It was my pleasure to read your work also. Congratulations for all your results and your manuscript. So maybe, yes, I can have several questions. So I would like to start with general questions. So your model is rather interesting because you have 4 different CRISPR subtypes inside the same model, so chromosome and also a megaplasmid. So I wanted to ask, do you think there are some sort of interplay between different types of CRISPR system? Because so you are presented figure with 1B, 1C, and 3 and 3B, and so sometimes we can see that you have this convergent transcription with different CRISPR-A, CRISPR7, 8 belonging to different types. And so do you think there are some links or interaction between different components, and why so many different systems here?
Yes, of course. I think that all actually defense systems in all organisms have some interplay between each other and so on. But if we speak about type 3A and type 3B systems, about their redundancy, I think that maybe from the first glance, it seems that these systems are very similar, but as my research shows, in fact, there are distinct features of all of them. And maybe, for example, 3A system relies more on the strategy that can be described as non-suicidal defense system, while type 3B, with the synthesis of cyclic oligodenolates and with the activation of different non-specific DNase, RNase, protease activities, might be rather lead to the cell dormancy than indeed survival of the cell. So I think that this is just 2 different types of immunity that allow cell to create a more strong arsenal against different phages, against different strategies for phages, because we also know that all phages have distinct life cycles. So maybe all the systems have different input against different phages. And if we speak about type 1 systems, they target DNA, for example. So it's like the first line of the defense that allows cell to coact against phage, but type 3 systems can be activated only when transcription starts. And it's mean that type 3 systems are more like last resort to the cell to survive. So all these systems have their unique features, and I think that all of them support each other, and because we don't know which particular auxiliary activities are activated by cyclic oligodenylates, and maybe there are some other second messengers, not only cyclic oligodenylates, and so on. So yes, I think that this is a very tricky, complicated network, and it would be great to know in detail how it works.
Yes, thank you. So do you think, is it frequent, the co-occurrence of different types of CRISPR-Cas? Have you in mind other examples?
Sorry, there was also some disconnection. So you asked about the, do I know some examples of coexistence of type 3, type 3, or just generally speaking?
Yes, generally, is it frequent to have different types coexisting?
Professor Stuturin, I'm afraid we lost you for several seconds again.
Yeah, I'm
Okay.
sorry for all this.
So I'm yes, repeat. So do you know if it
I
is?
understood your question, so I can try to move forward. I will not tell you exactly organisms and exactly systems, but of course I read about many of systems that can exist, and moreover, for example, type 3 systems usually don't have their arrays, so they rely on some CRISPR arrays belonging to another CRISPR system. So for me, CRISPR-Cas systems is something about modules that can be combined in different variations, and through the evolution we can see various examples of various mixtures of these modules.
Yes, thank you. So I wanted also to ask, so focusing on your work on type 3A and B comparison, and so you provided some results on mass spec, on the quantity of components, and so we also did something similar in our system. So I wondered, what are your opinion? So how you can explain these different amounts of proteins, and what are their hypotheses about this?
Yes, so in fact, when we first used our RNA-seq data, comparing infected and non-infected cells, we didn't see any drastic changes in the transcription of known Cas genes that are associated with type 3 immunity. But when we speak about proteomics, we also analyzed cultures without any additional immunity triggers, like without infection or something else. So these proteins comparing to other proteins were not so abundant, but still we see indeed differences between type 3b subunits and type 3a subunits, and type 3a subunits were detected better actually, but it doesn't mean that we don't have 3a subunits, type 3b subunits in vivo, because this procedure also has some limitations, and we cannot say that this is for sure the quantification analysis, let's say. I think that maybe the thing that type 3A system is more abundant is again the reason that type 3A system can trigger immunity that is not so suicidal as type 3B systems. So I think that 3B systems are more dangerous for cell, so it's better to keep this system under strong control. So maybe that's why we don't have the same abundance.
Yes, but they are both efficient for interference.
Yes, so we don't know the amount of the effector that is needed for the efficient immunity. That's why we see that immunity takes place. That means that even these amounts of protein is on the lower border for detection facilities, so still it's enough. Because actually type 3 systems are very fitness cost, has a high fitness cost for the cell, so maybe it's not a good idea to spend resources for all these defense systems.
Okay, and so do you know what are the mechanisms controlling the expression of these systems?
No,
If it is known.
Unfortunately, no. And as I mentioned, even during our RNA sequencing analysis, we didn't see drastical differences between non-infected and infected cells. However, we haven't checked resistant strains, so maybe in this case there would some interesting insights to analyze. So it's a very interesting thing to do.
Okay, so for future.
Yeah, there are so many directions actually that creates this system very interesting for analysis.
Yes, I agree. So maybe, yes, I had also some question on anticrease, but it's already asked.
We can't hear you. Could you please repeat from the very beginning? It's our problem.
Okay, yes, I repeat. So yes, I had also some questions, so it was rather interesting. Your candidate.
Once again, we can't hear you.
Oh.
Could you please repeat again?
Yes. So I'm repeating.
Yep.
So I was interested in your anti-CRISPR candidates. So, but Ed's already asked some questions, so I will not go through. So maybe, yes, I wanted to know, so in you mentioned here suicidal systems and also in your introduction, so, and you mentioned that for abortive infection, so they induce cell death. So do you think that it will be cell death in all cases, or it could be other scenario possible?
I think that, for example, in the case of type 3A systems, as I said, this system is more non-suicidal because actually, as in the previous discussion, were mentioned works by Luciano Raffini, and they showed that actually HD domain is the component that helps cells to recover from the dormancy created by the immunity. So, but in the case of type 3B system, I think that we don't have this HD domain. And if we speak about the independent action of type 3B system, I guess that it's completely suicidal because it caused because it mainly relies on the synthesis of cyclic oligodenylate. So I think that it's more suicidal, let's say.
Okay. So also maybe You mentioned during your presentation, also in your manuscript, these potential applications of your work for this editing of phages, and maybe also using this endogenous CRISPR system to edit, yes, your chromosome
I'm
in your
sorry,
model.
again, we
So
cannot
could you say
hear
more?
you for some reason. I think it's an issue from our side, so I'm very sorry for that.
Yes.
But can you please
Yes, I will repeat. So I wanted to ask about the potential applications. So you mentioned potential applications of your work for editing phages, also using endogenous CRISPR-Cas for editing genomes. Could you please say more about this?
Yes, so actually the genomic editing, especially in case of thermophilic phages, is quite complex thing, and we couldn't do it before. But for now, when we, for example, create spacers targeting transcript from the gene of interest, for example, and if it's, for example, non-essential gene, we can use this mini CRISPR arrays to perform gene knockout in phages, and only phages that bear these deletions will survive. But if we speak about more essential gene, that maybe we can use this procedure to show the codons that encode some amino acid that are not so important for the protein, viral protein. So, for example, in the case of RNA polymerase, when we target some other regions than they described in the manuscript, we cannot see any escapers, meaning that these regions are of high importance, for example. But still, even for our procedure with anti-CRISPR proteins, we can delete, for example, the protein number 42 is in fact the strong candidate, and we can use this mini CRISPR arrays to make deletions of this gene, and then we just can analyze if this phage can in fact cells with the same efficiency, for example, or maybe it will not be resistant for some other defense systems. So I think it's a really cool thing, and might be served for a good new insights.
Thank you. So I don't know if I have time, maybe one last question.
Yes, you do.
Okay, so yes, I would like to ask, so your particular system, so type 3, you have several activities. And so what do you think, what are the advantages to combine these different activities inside the same CRISPR-Cas?
Previously it was supposed that actually synthesis of cyclic oligodenylase is the most important part of type 3 immunity. And the action of HD domain is in fact not so important. But we showed that in fact it's not true for 100% because HD domain had a crucial role. However, no one saw in vivo cleavage by HD domain, so we cannot say what is the exact target for its activity. So it's quite difficult to make any speculations about this. But coming back to the third activity, the targeting of the transcript, in fact, this is a great control for the activity for cyclic oligodenylate production, because when this target is cleaved, then the synthesis of cyclic oligodenylate is abolished. That means that this let's say toxic maybe activity for cell might be stopped, and that's why cells can recover after all these things. But still, of course, if we speak about targeting some important transcripts for virus or for plasmids, it also has a great role because maybe it can give more time for other systems targeting DNA to provide resistance.
Okay, yes. Thank you very much for all the discussion.
Thank you very much for your questions. It was very interesting to discuss.
Thank you very much. Thank you very much. So we go now to Raymond Stahls. Are you with us, Raymond?
Yes, I am still here. Hopefully,
your turn to grill the defendant.
Yeah, thank you so much. Hopefully my connection is slightly better. I have to say that some of the questions I wanted to ask have already been asked by previous jury members, so I'll try to move my way around them. Before I go to my questions, I first want to congratulate you on such a wonderful...
I'm sorry, we cannot hear you.
Is it getting any better?
Yes.
Yeah.
Yeah. Yeah, it's difficult as it is. Now, I just want to congratulate you on such a fine thesis. It was really a pleasure for me to read, as you know.
Again.
Yeah, still troubles.
Technical support here. Can you do something about it?
I will try. I just wanted to congratulate on the nice thesis, and I really enjoyed reading it, especially since you did all the work that I was wanting to do myself. But I'm glad to see that you actually found the time to do it. So maybe I will just go to the questions before we have more interruptions. Like I said, some of the questions I wanted to ask have already been answered, so we'll try to skip over those. I do have some more general questions about type 3. So type 3 is very special, as you acknowledge yourself as well. It had a very elaborate network of different catalytic activities that happen. There's also a lot of differences between other CRISPR-Cas systems, and one of them is the use of a PAM. And many CRISPR-Cas systems use a PAM sequence to differentiate between immune, what is self and what is non-self. But in type 3, this works differently. However, there's also a theory that goes around that PAM sequences are there to quickly find the target, because otherwise the complexes will have to look at millions of sequences. Yeah, but
no, sorry, we didn't hear you. So you said about that PAM sequencing are important for taking spacers, and then we lost connection. So could you please?
Well, PAM sequences are also there to quickly find the target, right? So the complexes don't have to investigate every bit of sequence. But type 3 systems don't make use of that system. So how are they able, do you think, to find their target so quickly, apparently still?
Thank you. Yes, for the question, I think that maybe as during previous discussions, we said that type 1, for example, systems, they can be induced by some triggers and so on, but I guess that type 3 systems had to be present in the culture in the higher amount, so they indeed spend more time to find the target, but it was not shown that there are some connection with the RNA polymerase, for example. So the these complexes somehow are attracted by maybe some unbound DNA in the transcription bubble or something like this. So it helps the effector to come to the point where the action might be useful, let's say. And if they got this the protospacer, they can immediately turn on the immunity. So I guess there are some correlation with the target with RNA polymerase. So and as I mentioned that maybe for FIFA we could explain the different efficiency of spacers, because some of these protospacers might be transcribed by viral RNA polymerase. So I guess that there is a point of transcription.
Yeah, I might get back to that later on as well, but thanks for the answer. Another question is surrounding adaptation that I always wonder.
Lost you again. I'm very, very sorry.
So this is another question about.
No, we're here. Yeah.
Yeah. So the adaptation phase in type 3 is also very difficult, because in order for a new spacer to be effective, and you mentioned that as well, it has to be acquired from a region on the phage that's actively being transcribed, and B, it has to come from the right strand, because otherwise it doesn't work. You wrote part of the answer in your thesis by saying that, well, nature will select for functional spacers, because those that don't acquire functional spacers, they will die off. But it will sound like a very inefficient system to me, that it's kind of trial and error. And then there's some type 3 systems that have an RT domain fused to Cas1, but that's very minor subset of all the type 3 systems out there. So how do you see this? Is it really that ineffective way of randomly selecting a spacer and hopefully it's a functional one? Or how do you see this?
Yes, actually, as I mentioned, the first paper was devoted to the adaptation, and indeed we saw many spacers that corresponded to the genome, to the phage, to unfunctional spacers. Also, there were adaptation in to CRISPR-Ras, for example. So one of the spacers was functional, another was non-functional. So it seems indeed that this is not the most efficient system in adaptation, but maybe this strong adaptation itself creates a great resource for different spacers. So maybe it is faster but not so accurate. So maybe it's not so easy to say if it's efficient or not efficient when we're speaking about CRISPR-Cas system, because it's quite important to catch at least something to provide resistance against phages. If we speak about the same life cycle of phage, for example. So maybe this is the feature of type 3 system.
Yeah. Well, sometimes I think that also part of the answer might rely in the actual high transcription rates. High transcription bubbles is more likely to cause DNA breaks, and maybe therefore the Cas1 gets attracted. But that's for future studies to address. Another special thing about type 3 is the variable 3' ends of the guide RNAs or the CRISPR RNAs. You mentioned that also in your thesis. We developed a theory behind that, why that is, because it bugged me for over 10 years.
We lost you, I'm sorry. I'm not sure what is the reason, but I guess now we will hear.
Yeah, is it better now again? Yeah. Basically, my question, what do you think is the biological significance of having truncated 3' ends in the guide RNAs?
I think that it is also the question about the abundance of subunits, for example. So maybe if we have smaller amount of target, and for example we have longer complexes, maybe it gives some more time for a factor to produce the cyclic adenylase, for example, because we know that the cleavage goes from the tail, so maybe longer complexes will be more efficient in this way. For example, if we speak about shorter complexes, the efficiency will be enough if the target is the abundance of the target is sufficient. So maybe there are some correlations between the effects that can be possessed by the different effectors with different lengths. So I think this is some difficult, strongly orchestrated mechanism that somehow controls itself by these features.
Right, right. And all these mismatches that you were studying, I think they were chapter 5, do you think those lessons that you learned had a minimal, let's say, base pairing requirements between the CRISPR RNA and the target RNA? Do you think that will be different from differently sized complexes?
I'm sorry, I didn't get the final part of your question. So,
No problem. So the question is whether you think that shorter complexes will have a different mismatch tolerance than larger complexes.
I think yes, because, for example, if we just physically don't have the needed target that will be covered by the CSN3 subunits, for example, so of course we will not be able to see any immunity because the minimal length, for example, 26 base pairs, and if the subunits cover only, let's say, 21 position, that this complex will not be efficiently trigger immunity, I guess.
Yeah, I tend to agree, but it also means that the results that you depicted in chapter 5 are basically the average of all the different lengths of complexes that exist in terms. Do you agree, or?
Once again, can...
So the results that you showed on the mismatch tolerance in type 3, those results are basically then an average of all these different lengths of complexes, right?
Yes, maybe there is some explanation for the type 3 system, but I think that it's not true because I guess in your works you show that the main positions for activation is allocated at the 5' end of the CRISPR RNA. So it is like the head of the complex.
Yeah.
So I guess that since we see difference between type 3b and type 3a systems even in these regions, that means that generally speaking, type 3b system is more sensitive, and it's not about the general size of the complex. So I guess that maybe it's in opposite, because maybe HD activity depends more on this length.
Yeah.
So,
yeah, well, I think that's one of the highlights also of your thesis, that 3A is indeed very different, I think, than 3B. I've been mostly studying the 3B systems, and I always had the impression, well, 3A probably follows the same rules, but I guess the results in your thesis shows differently, so that was nice for me to see. Final questions. One of them is about the deletions that you found of the phage that were escaping type 3. And you showed some examples in your thesis. And some of these look to me like part of the protospacer is still intact.
we don't hear you, sorry.
It's about the deletions in the phage that you showed in your chapter. I was wondering, do you think that some of these protospacers, they are still intact? So they might still provoke RNase activity, but not HD activity. In other words, did you check whether the deletions that you saw in your phage, do they correspond to the rules that you learned in chapter 5?
Actually, if we, for example, come back to this slide, yes, they completely correspond to our results, and these deletions lead to the complete shift of the alignment. And indeed, when we saw so these regions that were present, let's say, in the protospacer, they were smaller than the region that we expected to provide efficient immunity. So yes, this data correlates.
Yeah, yeah. I agree with that. Then my final question was what you wrote in your last chapter.
Sorry again.
My last question is about your last chapter.
Yeah, now you're back. Thank you.
There you wrote that for your future plans, you were planning on making deletions in the CARF proteins, CAN and CSM6, CSX1, etc. Let's assume you have these deletions now. You have these strains. What are your plans on doing with them?
So actually, this is a project that is going on right now. And yes, first of all, we would like to check all this plasmid that we have with just starting from simply DIR and REF variants to check if for example, type 3b system will lose the protection. And since we don't know what genes, what proteins have the maximal involvement in these genes, we will just check different variations of the deletions and make the main players, let's say, that had a particularly input. Also, we will check if this is true for both 3A and 3B system, because as I said, we're not sure what type of cyclic oligodenylates, for example, are produced by these systems. Also, we don't know if cyclic oligodenylates in fact produced, because recently other types of second messengers were discovered about Malcolm Wall, for example. So maybe there are different sets of auxiliary proteins that serve for these systems, and this is the, I guess, primary idea, to check this one. And also we would try to predict maybe some more genes that are associated with type 3 immunity but were not described, for example.
Yeah, that sounds very exciting. Very good luck with those experiments. I'm very much looking forward to the results of that. I think that concludes the end of my questions. Thank you.
Thank you very much. I don't know if Robert Fagerlund with us. He's in New Zealand, and obviously it's mid of night. So probably we skip him, and if necessary, I will read his conclusions. Okay, so one of the roles of the defense is to disseminate knowledge among lay population, and I think I represent here one of the members of this population, so I will dare to ask a few questions, which I have not put into the thesis, which I agreed with the corrections, and this is just for my specific knowledge. So essentially, if I am correct, this CRISPR-Cas system which you studied is similar to Cas10, which possesses RNase activity, right? So the target of this Cas system which you study are RNA. Am I right?
Yes, yes.
In this case, my question is, Have you noticed any bystander effect similar to Cas10, where when activated with a specific RNA target, it starts to hydrolyze RNA molecules which are absolutely non-complementary to the sequence of cRNA. Do you know anything about that?
So we didn't check it particularly, but this question is complicated by the fact that our effector complex synthesize cyclic oligoadenylate that activate non-specific RNase activity of other proteins. So actually we didn't check.
But you mentioned that you've done RNA-seq in some of your systems.
Yes, but we didn't do it for strains that were resistant to the infections. So we checked strains, wild-type strains, and but again we made such experiments. In fact, and, we didn't get a good data right now, but I hope that maybe this is a direction that would be interesting to cover, so maybe we will find in this data something interesting.
Another thing which I just picked up from your excellent presentation is that one of the conclusions is that CAS system under investigation possesses recombinating activity because you found deletions in the plasmid. So it means that it should have DNase activity first, yeah? So which components of this system are involved in it? And then second question is, and it's probably more specific question, how do you prove that deletions which you see in the plasmids which you used with protospacers are caused by specific recombination, but not just by stochastic deletion from the plasmid, which are quite common if you have repeats in the plasmid. Do you understand the question?
Can I repeat it just to make sure that...
Okay, you have experimental result that your plasmids lose protospacers. Yeah, and you make the conclusion that this is the effect of A3B or A3A. So what is the proof that this is a specific effect and not just a serendipitous deletion of the plasmid?
So in fact, we didn't say that this is the reason of the activity of type 3A or the type 3B system, but we said that type, but we said that
recording in progress.
We said just that these cassettes arrays are combinogenic, and we don't go into the processes that are involved.
I see. I just overshoot. I reformulated it. So you are accurate. You say that the cassettes are combinogenic, not because my feeling was, and it was my mistake. Thank you for explaining it. So I think we finished with. Reviewers, and I do believe that all reviewers mentioned that they are completely satisfied with corrections to the thesis. Yeah, no answer is a positive answer. And now we ask the supervisor, Professor Siberinov, to say a few words about Karyna. Please, Konstantin.
Yeah, hi Yuri, and hi everyone, and thanks for taking part in this. Well, like they say, I never heard that actually being said by the Brits, but they say that Brits say the proof is in the pudding. So clearly, I think this is a good thesis, and Karyna has been working on it for, I don't know, almost 6 years, right? So she joined the group on type 3 immunity when she was just the second person you know, a studying student, master student. Now it's a much larger group, and we're exploring various aspects of type 3 immunity using thermos as a model. And Karyna not only graduated, or I hope will graduate in a few minutes, but she also started to be an essential member of that group because of all the experience of knowledge, practical knowledge, particularly that she accumulated. So she became a very, very essential member of the team, for which I'm very thankful, right? She's also a very good citizen and helps others to learn what she already knows. She didn't touch upon that, but she's also a phage hunter. So she participated in several expeditions where some of the phages, so in fact, the phages that she described to you are not the ones that she caught in the wild, but she did go to several remote islands off the coast of Sakhalin to bring some goodies in. And these were really nice phages. So I think, like everybody else, I want to congratulate her. I think she deserves, you know, the highest praise for the work that she did. She knows this. We discussed this. I think she can work on her presentation skills and also ability to succinctly answer questions. But I'm sure she will learn that and be better. She's good, but she can be better in this particular. But otherwise, I have no reservations.
Thank you very much. So we almost completed our schedule. Unfortunately, there was no questions from the audience.
Maybe we still have some.
You still have a chance. Yes, вопрос. Oh, please then. Please, the question from the audience.
Okay, no questions in Zoom, I can see, and questions from audience here.
Okay, then. So Konstantin was right. You should be a little bit more accurate when you say small cells, but this is small colonies and other small things. But this is a slip of a tongue, obviously, nervousness. Okay, then we're closed for the deliberations. Yeah. So we should organize a room in the Zoom, probably, Lena. And we ask the audience to leave, probably for a few minutes.
Exactly.
Thank you for being with us. I think it was another excellent thesis defense. It's my pleasure to congratulate you that you passed with options as is, so there were no minor changes requested. Philosophy, scientists. I wish you all the best in your future scientific career. It's just the start, a very important point of maximum freedom of choice. And I wish you that you make a good choice and you live long and successful life in science. Well done, Karyna.
So can I? Yeah. So all the audience online, please turn on your cameras. It will be much better if you can see in the photo. Guys, your present just turn on. Go. Hello. So, first of all, I would like to again say thank you to my supervisor, of course, for all these opportunities, for the guidance, for the mentorship. But also, I would like to say thank you for the jury members, for their kind words, for their comments, for fruitful discussions, and their time, of course, for viewing my thesis and making this event possible. But also, guys, I would like to thank all of you who are here today, who are online with us today. Thank you so much for your support. It's a really great thing to see you all and feel your support. support. Thank you again. Love you.